Jesus, thank you for the invitation to the talk today. It's nice to see all the interdisciplinary work going on here in Plymouth. My background is computer science in my undergrad. Then I went and did my PhD in the Manchester Interdisciplinary Biocentre. So it's quite in keeping with what you guys are doing here. But for today, I'm going to focus mainly on the work that I did in my PhD, which I finished about 18 months ago, and what I did in my first postdoc, which was in Singapore. And I'll touch on what I'm continuing there in Newcastle. So quite different to the previous two talks. I'll talk about brain connectivity, brain networks, and brain models. And so why would we want to model the brain? Well, it has applications in disease, such as epilepsy. And we're going to focus mainly on three particular applications in the context of epilepsy for the work that I've been doing. And this is particularly trying to understand brain stimulation, surgical resection of particular brain areas in epileptic patients to try and treat epilepsy, and also ultimately patient-specific modelling. So I'll give you a bit of background about epilepsy, motivate why we need to study this, and then describe some of the modelling tools that I've used in my PhD and beyond. And then I'll talk a little bit about brain networks and how the two can be combined. So there's various different types of epilepsy. One type that I've been particularly interested in is idiopathic generalized epilepsy. So idiopathic means that we don't necessarily know the source of the seizures. So it's not like someone's had a stroke and you can see it on an MRI. When you look at these MRIs of these patients, they look pretty normal. We don't really know what's causing the seizures. They're generalized in that they are spreading to at least both hemispheres of the brain, if not all brain areas. Epilepsy means recurring seizures. And IGE can essentially be considered an umbrella term under which many different types of epilepsy can be categorized. And for those of a nervous disposition, you might want to look away at this video at the next slide. Someone having a seizure, but it's kind of just to illustrate the point of how serious these seizures can be for patients. So here he's cognitively normal, he's awake, active, writing, and in a second he'll start to have a seizure. These are real people, real seizures. Tonics and subtension there. And the tonic-clonic first, so there's rapid tensing of muscle and then relaxation into these kind of autonomous organs. And the patient is cognitively not aware of what's going on right now. Obviously this is a serious condition. And these seizures are correlated with electrographic manifestations on the EEG. So the EEG is a piece of recording apparatus where we put electrodes on the surface of the scalp of the patient. And we can record essentially the electrical activity at that location in space over time. So here we have an example of this various different electrodes over the scalp. And these record in space, time. And when a patient has a seizure, we see a sudden change across, in this case, all electrodes, change from low amplitude activity to high amplitude activity, pretty correlated across most channels, although there is some difference in the spatial profile, so there's some spatial aspects that can be important. There's a change in the degree of synchrony as well. If you look at the synchrony between the channels here and the synchrony between the channels here, you will see a difference. And it's not really understood why these seizures happen a lot of the time. Like I said, they're idiopathic, so the MRIs look perfectly normal. They look perfectly normal, they're not necessarily are. And this is actually a surprisingly common problem. If you know 100 people, you probably know 1 or 2 people that have epilepsy. About 30% of those are inadequately treated, so drugs don't work to stop the seizures. They still continue to have seizures, even if they're on medication in this 30%. Or the drugs are so, that give such bad side effects that they worsen the seizures themselves. So this is a real problem for many, many people. In terms of the personal cost, it's obviously significant, but in financial cost, it's the most common cause of hospital readmission. So you'll see NHS a fortune to treat these patients. And like I said, the underlying reasons for this are poorly understood. We don't really know how to best treat epilepsy, if we're honest. Stimulation is one thing that's been proposed in recent years. Sometimes it works, sometimes it doesn't. We don't really know why. In this case, we have 2 seizures from the same patient. In the first seizure, I'm showing 1 electrode for place. But in the first seizure, essentially, a clap, an auditory stimulus stops the seizure, whereas the same in this seizure, it doesn't stop it. And we don't really know why it works sometimes and why it doesn't work other times. So if you ever see somebody having a seizure, try clapping, it might just work just on the off chance. For many patients, the medication is inadequate, and surgery doesn't always work either. So in the case where we have patients that are not idiopathic, where we think we know where the focus is, so we can see an abnormality in the MRI, a lot of the time what happens is they go into the clinic, cut away the scalp with essentially a circular saw. Sorry, we've got to go away the skull with a circular saw, put electrodes onto the brain surface, record that, and see if we can detect where the abnormal activity is coming from, even more localized, and then put that piece of brain tissue out. And it sounds brutal, but that's literally what they do. So hopefully that's motivated the problem for you. So some of the questions that people might ask is, well, I'm sure you can read, but I'll read a couple out. Where is the best place to stimulate to stop the seizures? So there's different places in the brain we can deliver the electric stimulus. Where can we stimulate? What's the best stimulation protocol? Is it 1 pulse at the top of a wave, is it another pulse at the start of the seizure? Is it 3 pulses, 2 cycles in? Where's the best place to operate? Is it where we think that it's starting, or is it somewhere else where we don't necessarily know where the seizure's starting? If I can't operate here, where else might have a good outcome? So I think this is an under-asked question quite often. So a lot of the time you'll see an abnormality on the MRI and you think, right, we can operate there, and then we realise that that's where focus area is, so that's responsible for language. So if we operate there, yeah, we'll stop the seizures, but they won't be able to speak anymore, so that's bad. So we can't operate there, so what do? Why does some surgery sometimes work and sometimes not? So it's kind of the rule of thirds, it's weird, but in about a third of patients where surgery is done, it doesn't work in terms of stopping seizures. So can we make a guess before we do the surgery whether that's going to work or not at all in this particular patient? So in terms of making guesses about the future, I propose that we should use computational models. So computational models are quite often used in many things that you might be aware of. Weather prediction, the forecast for today will actually rain, worked. We didn't necessarily see the stock market crash come in, but they are used computational models there. A friend of mine doing a PhD at the same time as me did modelling as well, and he works for Auto Trader, which is the biggest second-hand car retail company in Britain, I think. And he got a job doing models of car prices, predicting car prices, which is quite cool, I thought. And there's loads of other examples. And so the goal really of my work and my presentation that we're going to talk about today is, can we make predictions of optimal treatments for patients that are clinically relevant? So not predictions just for the sake of it, but are clinically relevant. And so, like I said, I'll focus on 3 particular problems. The brain stimulation work I'll talk about is quite long. The surgery prediction work is quite short, and then patient-specific modelling stuff is kind of going to be the end of the talk. So if you're getting bored halfway through, know that there's another section coming up. So just to show this slide again in the context of brain stimulation, we've got a few questions that we might want to ask. And additionally, what are the potential drawbacks of doing this stimulation in a particular patient, for example? So I'm going to talk very briefly about the model, because I think there's a few people that are not experienced with modelling, and I don't want to dwell too much on the equations. So in this case, we model the cortex and the thalamus, so the thalamocortical loop essentially, because we know from experimental evidence from Steriadi and Dash from about 20 years ago that this loop is important in epilepsy, particularly idiopathic generalized epilepsy. And we have thalamocortical and reticular neural populations, pyramidal neurons and inhibitory interneurons, and some sort of connectivity between them, which has been shown experimentally that these connections do exist. And we use a set of population models similar to Wilson, Kevin and Mary type models for anybody that's familiar with them. And we neglect for the time being lateral spatial interactions. So because we consider the whole cortex to be kind of synchronized, we neglect different cortical locations for the time being in this particular part of my talk. So that's the model. I should say this is the noise-free case of this model. If we add noise onto this model, then it's essentially that plus a noise term at the end on an ITSD. And so when we simulate the model, we get transitions between a background state, which is low in amplitude, and the seizure state. Yeah, you can see it. And so we have a spike wave in the simulated model, and this compares quite well to the case in the actual patient. So we have this kind of fairly sudden transition to the spike wave, sudden transition back again, 10, 12 seconds. If you do a power spectrum on this for the background and the seizure, it's comparable as well in terms of background alpha beta activity. And seizure is at the delta 435 dose, about 2.5, 3 dose. And that's because we have a bistability in the model. So this slide is going to look hideously complicated to a lot of you. So the main thing I want you to take away from this slide is that there's a bistability in this model. So the bistability means that there are two stable states that co-occur at the same time. The only difference that decides where you end up, like which state you end up on, is due to a perturbation away from one state to another. So that perturbation can either be, for example, a stimulus, or it can be noise, which is essentially a time-varying perturbation. And I'll not dwell too much of it on this, but this is kind of perhaps the most useful thing to try and understand what a bistability is. So we have the different variables, the different populations in the brain, so the pyramidal neurons, the particular neurons in the thalamus, and the thalamocortical neurons. And when we plot the spike wave of each variable, we essentially get this, what's called a living cycle. And so in the noise-free case, this just continues indefinitely, and it's always identical. And we also have this co-occurring with a stable focus, which is a fixed point in the middle. And in between the two, we have what's called a separatrix, so a separating manifold, which kind of, if you start off on here and you have noise, if the noise is not sufficient to drive you beyond the separatrix, you'll stay in this region. If the noise is sufficient to drive you beyond the separatrix, you go to this part of phase space. You do the limit cycle and back again.
So you said you have alpha and beta, where does the same frequency go from?
So because it's the focus and not a node, and there's noise added on, you get a kind of resonance around the focus. That's widely used. That's widely used for modeling background, there's noise and stable focus. So the key thing to take away from this slide is that it's a bistability in the system. And so this is kind of plotting through the TC and RE plane, if you like. And we start off on a fixed point and we could give stimulus away from that. Like I say, if you stimulate to anywhere in this green area, we'll always return back to it. If you stimulate to anywhere in the black area, will result in being a spike wave attractor. And that's because it's bistable. And so this is important because if we're already on the spike wave attractor, and what we want to do clinically is to stimulate away from the spike wave attractor to the background attractor. And so if we're already on it, then we need to stimulate into this region, if that makes sense. So this is deterministic, so there's no noise in this. When we add noise, interestingly, although the same sort of structure is preserved, we get a variation from trial to trial depending upon what the noise is.
Is it based on the attraction?
Yeah.
But you have more variables, what another one?
Yeah, so this is essentially just a slice through two for visualization. I'll go on to more variables in the next slides. We have, when we add noise, we get variation from trial to trial. And interestingly, we get some structures due to the noise kind of go on, and we have a probability of the success stimulating back to the background. So the probability is a number between 0 and 1, if you like. It's not a binary, yes it will go back or no it won't. The probability of going back. Because the noise can always kick you back out again, even if you stimulate into the background. So that's our first key result really, from a model that gives us a prediction that noise will cause variations in success from one trial to the next. And if you remember from the original, I showed a few slides ago from the clinical observation, there was variation in success between those trials. But what other factors can play a role? So I'll start off on that one, this bit here. We can essentially scale the time at which we apply the stimulus perturbation for a fixed amplitude. You see, if we stimulate here, it doesn't stop the seizure. And if we stimulate here in the green area, it does stop the seizure. This is essentially because we're stimulating into the basin of attraction. So this is the basin of attraction in 3 variables, the PY, the TC, and the IN. And this is showing the basin of attraction with the PY, TC, IN, with the 4th variable RE mapped onto time. So this is a static 4-dimensional structure viewed. It looks like it's time variant, but it's actually not. It's fixed in time, but it varies because of the 4th variable, depending on where you're stimulating. So here, the reason that this one works basically is because you're stimulating into the background basin of attraction. So that's like the green area on the previous slide. So the green area is not limited to that nice circle when you look at it in more than 2 variables. It's actually ridiculously complicated. And so when we stimulate into the background basin of attraction, at this time it doesn't work, and at this time, which is essentially the same amplitude. So a decrease in IN and a decrease in PY. So a decrease in PY and a decrease in IN. It's the same amplitude in the same direction. It doesn't work because the basin of attraction is essentially moved in a way, even though it's a static structure, because of the value of RE changing. And so it's strongly dependent upon the timing at which we apply the pulse, and obviously following on from this, it's also dependent strongly on the amplitude of the pulse. So when we stimulate here into the basin of attraction, yeah, great, that stops the seizure. If we stimulate here, even stronger, it's not going to work. So there's a strong dependency on the amplitude as well. So you might think intuitively, we try stimulating, it doesn't work. Try stimulating a bit harder, it does work. You might think, oh great, we just need to stimulate really hard. That's not the case, as predicted by the model at least. Because you get this kind of discrepancy where you can have it working, it not working, it working again, for example, on the bottom here, for different amplitudes. This is a bigger amplitude in the other hand. So it's highly complicated, even in just 4 variables. Obviously, they break to more than 4 variables, but we can at least make attempts try and understand this in 4 variables. So the key result from that slide is that there is a strong dependency on the phase, or the timing, and there's also a strong dependency on the strength, amplitude, as to whether the stimulus is going to work. And also, follow on from that is the directionality. But if we assume that we have had stimulus to both variables at the same time and the same amplitude, then it's going to be the same direction that we thought. So that's the second key result from the stimulation. So we've essentially so far highlighted a few factors that can contribute to the understanding of why stimulations sometimes work and sometimes don't work in patients. The final one that I want to show is, so essentially on that last slide, I showed here this map, essentially of stimulus amplitude against stimulus time. And in the deterministic case where there's no noise, this map is always identical from one cycle to the next because it always follows the same cycle because there's no noise to move it away anywhere. Whereas when you have noise, you get variations from one cycle to the next. So you might see this cycle here has a slightly lower amplitude than this cycle here. And that's because the noise is essentially moving it around in phase space. And as it moves in phase space, it becomes closer or further away from the background basin of attraction. So the noise can cause variations in the degree of success due to cycle variations. So this is important because this is a different mechanism to the initial noise result, although it's another effect of the noise. And so this is a reconstructed phase space in our model, and this matches well with human EEG. So essentially what we're saying ultimately in our work is that when you're doing these stimulations, you can't just arbitrarily stimulate because it's not necessarily going to work. You need to consider the phase-space structures, and you can do this through phase-space reconstruction from the actual patient data. So to get back to my original questions of why does stimulating only sometimes work, we're given three categorical mechanisms as to why they might or might not work, which will give variation from child to child. Can we predict if it will work? With absolute certainty, no, we cannot. I believe if there is noise in the brain, if we believe there's noise in the brain, then no, we can. We can predict the likelihood of it if it will work, but we cannot predict with certainty if it will work. The noise can always kick you back into the seizure state. What potential drawbacks are there to stimulation? Well, one potential drawback is if the stimulus kicks the limit cycle further away from the background basin of attraction, then potentially it might take longer to return back to it in the presence of noise anyway. So here we use the same noise vector but we stimulate at different times. So here is the original seizure. Here we stimulate it this time and it doesn't really have much effect. Here we stimulate it at this time and it prolongs the seizure, and here it works. And that's not just because we stimulated earlier, it's because we stimulated at a different time, which is at a different phase, in a different location in phase space. And this has actually been seen experimentally in rats, that you can stimulate to prolonged oscillations at least. I think warning. So this is kind of a summary slide for the brain conversion section. So if you didn't understand that, don't worry, I'll get on to the next one. Basically, there's no single pulse that will always work, I believe, if there's noise. These are the three factors that explain why. Stimulation epilepsy is extremely difficult. That's why we need something more advanced. That's why I think it's not really been working so far. There's been plenty of experimental studies that are trying to do it, and none of them have had proper conclusive success, in my opinion, in terms of single pulse stimulation. And future studies, I think, should probably be based on patient connectivity. So at the moment, I've just looked at a single location in the cortex. So it would be good to extend this to multiple spatial locations and see where should we stimulate, in addition to the three mechanisms for when should we stimulate that I just described. But more about that later. We'll get if you're interested. So the next one is surgery that I'll talk about. This is very preliminary work, and I'm just going to skim over it really. It's a student we had in Singapore, and it's his work mainly. He did really well. His work that I've seen in this context. So if you put a reminder of some of the questions, where should we operate? If we can't operate there, where else should we operate? And will it even work at all? So in this model we also have biastability, although it's a lot simpler this time. I'll not bore you with details of the equations, but we have a limit cycle around an unstable limit cycle, so a stable limit cycle around an unstable limit cycle, and then a focus in the middle. And you run noise to it. Kind of occasional transitions from low amplitude to high amplitude oscillations, and you can compare these to seizures, similar to seizures if you like these. And this was originally proposed by Celine Collective. This is not my model, I'm the group using his model. And followed up by John Terry in a couple of papers. Essentially what we're doing is we take an individual node to be each one of these bistable oscillators, and we couple these nodes together based on patient connectivity data which we infer from this kind of data. And we say, right, in the model, which nodes have a seizure more often than other nodes. We'll measure the escape time, so the amount of time it takes to escape into a seizure. And then we ask the question, are those nodes in the actual epileptic focus? So I should just say that we use interictal data, so non-seizure data, to infer our model. So we have no clue as to whether we're blinded as to what the actual seizure looks like, which makes this quite nice. So the nodes in grey are where the clinician, the surgeon, thinks that the seizure focus is, and that's where he actually operates it on. Nodes in green is where our model predicts the focus is. So these nodes in the model have a shorter escape time than other nodes. And then the next question is, if we reset those nodes, will that lead to an increase in the average escape time of the other nodes? So here's the escape time of the nodes before we do surgery. Here's the escape time from the nodes after we do simulated surgery. And here's the escape time if we repeat that for just a random node. And we see that by resetting these nodes, we make a prediction that the surgery will work. More so than if you resetted random nodes at chance. So that was quite nice. I think this particular patient did have a successful outcome. And I guess one of the predictions that we made is perhaps the surgeon didn't need to reset these nodes in order to result in seizure freedom for the patient. And we did that for a few other patients. Like I said, these are preliminary results. We're working on a follow-up study with many more patients. And there's some nice agreements here, so I think this patient was quite interesting. That the surgeon just suggested this particular node was where the focus was, and we got that basically. We also found this area, and so if this area is, for example, language, and the surgeon says, right, we can't operate there, what should we do? The model essentially predicts that if you operate there, you might have some degree of success for the patient.
What is the difference in your model between two different models?
So it's homogeneous parameters for heterogeneous connectivity. The only difference between each node is the connectivity, which is inferred from the back range equilibrium values.
Node is 4 equations, and they are connected according to...
So each node is 2 equations.
Yes, it's either that
Yeah, essentially.
connect them like nearest connection.
Yeah. Well, they're connected according to the functional correlation between the time series provided in the code from the patient. And yeah, you can see the other results for yourself. I thought the top one was quite interesting. We did get one case where it didn't work, but I can't remember the outcome. We will just talk about collaborations actually. People should, the clinical collaborators should go on that course that you mentioned, because it's a nightmare to get follow-up data from surgery on patients. Did the surgery actually work? I don't know. But in the one on previous slide, I think it did. So that's the summary for this section. It's quite short. So where should we operate? We make predictions made in green. We might operate there. Where else might work? Other regions with short escape times. And will surgery work at all? So we need to work on this still. But we want to compare retrospectively to the patient outcomes. So we need to go back to our collaborators in Boston and say, did it work in these patients? And simulate what they actually did. So that's what we're on at the moment. And finally, patient-specific models. So every patient is different. Everybody has a different brain. With evidence that seizures are stereotypic, so if one seizure is more similar to the next in the same patient than it is to a seizure in a different patient. And so if we have, especially in the case of IgE, where there's stereotypic properties which are distributed in large brain networks, then we should incorporate large brain networks into our model, I think. And so one might ask, how do we get a large-scale brain network of a patient? Well, we can do this kind of pipeline, and I'll just talk you through briefly how we get a network like this for a particular patient. So you've probably all heard of MRI, scanner type thing, big magnet, put a patient in, picture of the brain. You can also do DTI, which measures the diffusion of the water in the brain, which I'll talk about in a second. So the first step in this pipeline to get from scans to network is called parcellation. So essentially we segment the brain, so we trace the outline of it between the grey matter and the white matter, and we inflate it, match it to a pre-existing atlas, and then we say that this region is the frontotemporal region, and the borders are the connect out, so we replace it. Which doesn't really come in. And then we have a patient set of nodes which are correlated with an existing atlas. That is the probabilistic benefit. So this is getting the shiny pictures that you were talking about earlier. So in each voxel you can measure the diffusion of water. So if it's isotropic, and it diffuses in all directions equally. If it's anisotropic, then it's restricted by something. So for example, an atom bundle. So in this case, it's restricted by atom bundle, so the water doesn't diffuse that way as much as it does that way. So because there's an atom in the way. So we can do this for each and every voxel, and essentially we just use a tractography algorithm to track from one voxel to the next, and then we get networks of streamlines which are inferred from actual patients in terms of their brain connectivity. And then the next step is to align the data. So we have the cortical parcellation and the DTI. We want to combine the two so that they're in the same space. So we need to essentially put them exactly on top of each other so that we don't get any mismatches. So it's just image registration and get rid of motion correction problems. And the next step is essentially to combine the two to then infer the network. Thankfully, there's a nice toolbox to do this. It gives a big citation things there. It's very useful. And we've got nodes on one axis, the same nodes on the other axis, and each value in this matrix represents the connectivity. So the number of fibers, for example, and across the diagonals we've got cell-to-cell connectivity. And that's just repeating what I just said in a simple way. And so we can use neural population models to connect these different nodes. So just like I described earlier, like the Collectin model of, for example, the Carina or Maori type population models, this macroscopic scale. And then we can directly incorporate the connectivity matrix into the equations. So here's the equations, you don't need to understand them. We'll get excited during integration population neurons. We've got connectivity matrix directly in this equation. So when we simulate it, we're including the subject-specific connectivity. This is actually a model based on real human connectivity. And just think about that. In this case, I did a hierarchical scheme, which is a bit complicated than here. This is kind of two scales, so local connectivity and then long range. And then, so that's just a reminder of what an actual patient seizure looks like. And this is what we get when we simulate the model. There we go. And it's a pretty good match in many ways. So you get the slight wave oscillations, the slowing down between the different background, the rapid generalization across many channels and offset. You get more dominance in the frontal channel, so if you want to Z, if you want to Z, which is observed in patients. So the models reproduce many aspects of actual patient data that we see. So this is kind of just to whet your appetite in a way as to what these things can do. Ultimately, I'm going to combine this with the two previous approaches to try and better predict patient outcomes for surgeries and for stimulation. And that's just an overview of the pipeline. You take the data, incorporate the model, simulate the model, and then compare that with the actual patient observations. So to summarise my whole talk, computational models can be used to better understand the patient outcomes, I believe, just like we can use computational models to better predict the weather. Patient-specific data can be incorporated directly into models. We have video imaging modalities such as DTI, diffusion to injury that is. And there's a few groups out there that already combine models and connectivity to try and understand brain function, but there's a lot less that do this to try and understand brain dysfunction, so epilepsy, Parkinson's, things like that. And I think that that's an emerging field, to be honest. So, acknowledgements to my boss, my old boss, my older boss, and clinicians who do the real work. And thank you for listening.
Simulate the kind of seizures those patients have as well?
Yeah, this is basically the paper I showed at the bottom. I compare the similarity from one seizure to the next within a subject, and one seizure to the next between subjects, and I found that it's more similar to seizures are more similar. And basically reproduce the stereotypes that we see. That's kind of a hint towards the future directions. Developing the model is a hell of a lot of work in the first place, but now it's up and running. We hope to do good simulations in terms of surgical resection and practicing stimulation for skills.
Questions?
Impressive and important work, thank you. What happens to your model, and equally like for a patient, if it has been done, if a constant field potential is given to a part of the network?
Constant field?
Yeah, so do you know what a local field potential is? You can give a group of cells another field potential, and then favoring, you can prime the brain, for example, by doing that, part of the brain. What if you could do that with part of the network or the whole brain in your model and in a patient, or did you look at it and is it not done in patients as well?
One thing you could do to investigate that would be to apply a stimulus to one region and another region and see if they form a connection between them. For example, neuroplasticity, I'm thinking about it there. I don't include neuroplasticity in the model at the moment.
And I'm particularly thinking may that take away the biostability or in the patient. And not having the biostability may help in, or very much biasing is, it may help in the frequency of seizures.
Yeah, so in terms of the frequency of seizures, do you mean how often the seizures occur in a patient?
Yeah.
Right. So there are, somebody's done a study, Suchinsky, and he looked at how often seizures occur and how long those seizures are. And he showed that this follows a particular distribution which correlates with biostability in IgE patients. And he also did that for patients with temporal lobe epilepsy, TLE, and he found that followed a different distributions. So he suggests in that paper that patients with TLE have essentially a parameter on a slow change that's going on in the brain, whereas patients with absence of seizures and IgE essentially have a biostability that exists, or something in the parameter that's incredibly fast. I did that.
And that will also follow from your model?
Yes, because it would follow that distribution. I do actually have, in my PhD thesis, I put his figures with experimental distributions, I put the distributions for my model. Make sure we got that. It's definitely a fair point, because this does change on a patient-specific level. So we could potentially infer the distance from the two stable structures, or the distance of the separatrix between the two stable structures on a patient-specific level, which would certainly be very interesting. One thing that just helps me answer the question. Sorry about this. Get in there. There we go. So, is that all now?
Epileptic seizure.
Anyway, so the one where I had the massive lung time series at the top, that was simulating 6 hours. So I did that. So I did that for 36 hours until the distribution of seizures. And you can see on that time series if it came on.
I go after that word, thank you.
Yeah.
Yeah.
Great talk. You talk about modeling the brain using structural connectivity techniques like DBA. Do you think it will be more useful to use functional connectivity approaches like resting state fMRI for modeling the brain? Because especially in case of epilepsy, it's usually the function which is disrupted rather than the structural connections.
Very good question. I think that we should use DTI to inform the model connectivity and then simulate fMRI and then match that with the fMRI from the patient. So there are equations that you can kind of bolt onto these Amari-Wilson-Cohen type equations, which are called the balloon model, and kind of just go on top of it in a way, and that simulates the hemodynamic response function, and so you can do simulations of fMRI from these kinds of models. I'm yet to do that, but it's a very good point to do so. But it should be a model output rather than a model input. Another point on that is that the patient, in terms of getting that data for when the patient is seizing, for convulsive seizures is nigh on impossible because they're seizing inside the scanner, so you can't get fMRI recordings of people seizing unless it's an absent seizure. So that's an issue with that.
As I understand, your stimulus means you apply some level of current or potential and keep it for some time. Is that correct?
Yeah, essentially the way we simulate it is to move it somewhere else in the phase space.
Yeah, but say in deep brain simulation, people use periodic simulation with high frequency, and they found it's not very effective for epilepsy. It's not very clear. Sometimes yes, sometimes not. Can you comment on that? If we stimulate like periodic signal and we vary frequency.
Yeah, so we could simulate that, and it would possibly just bounce it around, so it might stimulate it in and then bounce it, stimulate it back out again of the pace of projection. It depends on when you do it and what the directionality of it is.
No, you need to think about additional dynamical regime.
I mean, you would model the stimulus as another variable, probably.
Okay, we want to hear that.
Sorry, just to follow up on what you asked, you say it bounces it around, but can you not just check to see if it's worked in between? I found myself sitting here thinking, you don't know whether it's going to help or hinder, but if you, do you see what I mean? You give a stimulation to see if it works.
Yeah.
So why would you carry on giving that series if it has worked? Can't you just repeatedly stimulate until it does?
I guess you could do, but you're then running the risk of damaging it. It's more of a rotate body variable if you just do it with a single pulse that's targeted.
Sure, but if you don't know how to do it with a single pulse. Sorry, that was my question. And you just asked something very similar, so.
Fair point.
You simulate the epileptic oscillations as the interaction between excitatory and inhibitory population. Is it actually known that that is the cause of epilepsy? I've seen many models like that, but I never heard anybody saying that is what has been observed.
So in terms of the cause or a consequence, I don't think anyone could definitively say. But one thing is for sure, whenever somebody has a seizure, there is always an electrographic manifestation of that. So it's a fair assumption to say that we should incorporate that into the model.
But it could just be the excitatory cells that synchronized without engagement of the inhibitory cells. That would be other reasons.
But if there's excitatory cells synchronizing them, there's probably something that's driving them down in order to drive the oscillation.
It's
So
just
one
to
would
get
get
exposed.
what is there to drive them down.
Well, the point is I never really saw evidence, biological evidence that it is a correlation constellation between excitatory and inhibitory cells, so I thought you might know something.
Yeah, so I mean, there is certainly differences in terms of the inhibitory firing, excitatory neural firing during the seizures. I mean, they both fire to some extent. There's different points in the oscillation. But in terms of your original question of whether that's the cause or a consequence of something else going on in the brain, I think that's unclear, and I wouldn't want to commit to saying that that's the cause.
Yeah, questions?
I was just wondering if you could comment on any animal work that might tie into what you're doing?
Yep. So there's a paper by into the bee. He did it in Science last year, and he wrote Rhythms of the Brain, the book. I can't remember his name. Was that? That's the cat.
Okay, I'm going to break us now for lunch,
Okay.
but I think a lot of people like to talk to you. So let's carry on at 2 o'clock.